Some traits are coded for by more than one allele, and so we call these multiple alleles. Examples include fruit flies' eye color, the color in rabbits, and blood types. For each trait observed by Mendel, only two alleles existed, capital T and lower t, for example, so tall and short for the pea plants. Many traits are controlled by multiple alleles. Multiple alleles are a gene that has more than two alleles. So we have eye color in Drosophila, blood types in humans, and fur color in rabbits. 4 alleles control the eye color in fruit flies, or Drosophila. E1 is wild type. They have red eyes. E2 codes for apricot-colored eyes. E3 produces honey-colored eyes. And E4 codes for white-colored eyes. The order of dominance is E1 is most dominant, then E2, then E3, and then E4. So this means if they have an E1 in their genotype, they automatically have wild type or red-colored eyes. Let's make a list of the possible genotypes for wild type. So they could have E1, E1, and we know that if they have an E1, they can have any other number. So E1, E2 would also produce wild type eyes, E1, E3, or E1, E4. In order to have apricot eyes, they cannot have an E1 in their genotype because they would not have apricot. They would have wild type eyes. So apricot, they can start with E2, E2. They can have E2, E3, or E2, E4. For honey, they can either have E3, E3, or E3, E4. And last, the only way to have white-colored eyes is to be recessive, so to have E4, E4. Let's try a problem. Predict the phenotypic outcomes if a honey-colored heterozygous fly is crossed with a white fly. So we'll put the gametes for these parents into the Punnett square. So E3, E4 on one side, E4, E4 on the other side. And we see their offspring are either going to be honey or white. Now there are 2 and 2 in the Punnett square, but when we're dealing with ratios, it's similar to fractions. We always put it in lowest terms. So for every 1 honey fly, we get 1 white fly. And so we write it as a 1-to-1 ratio. Genotypes is also a 1-to-1 ratio. Let's try this one. What are the possible phenotypes of the parents in a cross which produces the following offspring? And the Punnett square is filled in. So when we're writing the alleles for the parents, for example here, the allele has to be present in both this offspring and this offspring. And we can see that E1 is here and E1 is there. So that allele would be E1. Here we have E3 and E3. So that's E3. Here there's an E2 and another E2. And finally, here we have E3 and E3. So that must be E3, which means the parents' genotypes are E1, E3, and E2, E3. And the parents' phenotypes, one is wild and one is apricot. Rabbits also have multiple alleles that code for their coat color. We can see gray has a capital C. It is most dominant. Next in line is chinchilla. That is a color of rabbit. And then Himalayan, and then albino. And you notice there's no one color or one allele that codes for light gray. It's a combination of chinchilla and Himalayan, or chinchilla and albino, that produces a light gray color. So on an exam or quiz, you'd be given this entire chart. You don't have to memorize any of the genotypes for rabbits. Another example of multiple alleles are blood types. We have 3 alleles for blood type. It is the surface antigen on the red blood cell that determines blood type. It's an example of multiple alleles, and the 3 alleles are capital IA, capital IB, and little I. Little I codes for type O blood. IA and IB are said to be codominant because when you inherit both of them, you have AB blood. One is not dominant to the other. They are both present on the red blood cells. So if a person inherits both of them, then antigen A and antigen B are on the red blood cells and will be blood type AB. Little I is recessive to IA and IB, which means the only way to have type O blood is to have little I as the genotype. Let's write this down in a table. So have type To have type A blood, a person could have IA, so homozygous, or since IA is dominant to little I, they could also have IAI, so they are a carrier for type O blood. Type B is similar. They can have IB or IB little I. Type AB has to have IAIB, and type O is recessive, so it has to have 2 little i's. Let's try a problem. Predict the phenotypic outcome of the offspring for a person with blood type AB and a person with type O blood. So one person has the gametes IA and IB, and the other parent is going to have little i's for their gametes. If we cross this, we find that half of the offspring have type A and half are type B. So it's 1 type A to 1 type B, or genotypically also 1-to-1 ratio. Okay, if we are given the blood type of the mom and baby and unknown dad, in order to find the genotypes, we'll write down the possibilities. So the mom could have homozygous type A blood or heterozygous. The baby has blood type B. So we know the baby has a IB in the genotype and could be IBIB or IBI. Let's look between these two. The mom has to have an IAI in order to give the baby the I. So mom is not IAIA and baby is not IBIB, which means dad has to have a B in his genotype. So the dad must have an IB, so his genotype could be IBIB, IBI, or IAIB. Let's try another blood typing problem. This time mom is blood type B, baby is blood type O, so I will write down the 2 types of blood for mom, but we know she has to give the little I to the baby in order for the baby to have 2 little i's in its genotype. Dad also has to have a little I to give to the baby, so could be IBI, IAI, or have type O blood, 2 little i's. Calculating probability in genetics. In genetics, it is often useful to determine the probability or chances of an event occurring. Remember that previous events have no effect on the future events. Just because you have 3 daughters does not mean that you will have 100% chance of having a 4th daughter. The chances of having a daughter is still 50-50. So 50% chance of having a boy and 50% chance of having a girl. Probability is the number of chances for an event divided by the number of possible combinations. So in the chances of boy and girl, it's either boy or girl, so that's 1 chance out of 2, boy or girl. The product rule. We use this to calculate the probability of 2 events occurring simultaneously. So we multiply the probability of event 1 times the probability of event 2. In this unit, we usually find the probability of, let's say, blood type or eye color or anything, and then times it by the chance of either having a boy or a girl. So this probability of event 2 is always 50% chance, so you would multiply it by 0.5. Let's try a question. Calculate the probability of producing a son who's blood type A from the following cross. So we have one parent who's IAIB, the other parent is type O. We'll find the chance of having type A blood first, and that is 0.5. We also know the chance of having a son is 0.5, so we multiply those together, 0.5 to times 0.5. Final answer, 0.25 chance of having a son who is blood type A. Let's try another one. Calculate the probability of producing a daughter who is blood type A from a cross between a type B mother and a type A father, given that each of these individuals have a parent with type O blood. Because they have a parent with type O, we know that both of them have a little I in their genotype. So the mother must be IBI and the father must be IAI. So we'll put their gametes into the Punnett square and find the possibility of type, we want daughter who is blood type A. So type A is right here, 1 out of 4 chance, so 0.25. And we're going to times that by the chance of having a daughter. So 0.25 times 0.5 is 0.125, which we will round to the nearest hundredth for a final answer of 0.13 chance.